Hey, you are watching AliAziz YouTube channel.
ZN, Retic and Trichrome staining techniques. AliAzizQualityEducation YouTube channel provided you the medical related topics videos. So don't forget the subscription button. In histology, the art of staining is a fundamental practice that brings the invisible world of cells and tissues into vivid clarity. Among the many staining techniques available, ZN, Ziehl-Neelsen, Retic, and Trichrome staining hold special places for their ability to reveal specific structures and components within tissue samples. Each technique has unique applications and provides valuable insights into different aspects of tissue pathology. This article delves into the nuances of ZN, Retic, and Trichrome staining, highlighting their purposes, methodologies, and the human touch involved in their application. ZN staining, Ziehl-Neelsen staining. Purpose and application. The Ziehl-Neelsen, ZN, staining technique is primarily used to identify acid-fast bacteria, such as Mycobacterium tuberculosis, the causative agent of tuberculosis. This stain is crucial in microbiology and pathology for diagnosing infections caused by mycobacteria and other acid-fast organisms. Principle: ZN staining exploits the unique cell wall properties of acid-fast bacteria. These organisms have a high lipid content in their cell walls, which makes them resistant to decolorization by acids. The staining process involves the following steps. Primary stain. The tissue sample is first stained with carbolfuchsin, a red dye that penetrates the waxy cell walls of acid-fast bacteria. Decolorization. The slide is then treated with an acid-alcohol solution. Non-acid-fast organisms lose the red stain and become colorless, while acid-fast bacteria retain the red color. Counterstaining. To provide contrast, the slide is counterstained with methylene blue, which stains the background tissue and non-acid-fast bacteria blue. Key features. Visualization. Acid-fast bacteria appear as bright red or pink rods against a blue background, making them easily identifiable. Diagnostic use. ZN staining is a gold standard for diagnosing tuberculosis and other mycobacterial infections. Its specificity for acid-fast organisms makes it an indispensable tool in clinical microbiology. Retic staining, reticulin staining. Purpose and application. Reticulin staining, often referred to simply as retic staining, is used to visualize reticular fibers in tissues. These fibers are a type of collagen that forms a delicate network supporting the cellular components of various organs. Retic staining is particularly valuable in assessing the architecture of the liver, spleen, and bone marrow. Principle: the retic stain relies on the affinity of silver ions for the reticular fibers. The process typically involves oxidation. The tissue section is first oxidized to enhance the visibility of reticular fibers. Silver impregnation. The section is then treated with an ammoniacal silver solution, which binds to the reticulin fibers. Reduction. The bound silver ions are reduced to metallic silver, rendering the reticulin fibers visible as black or dark brown structures. Key features. Visualization. Reticulin fibers appear as thin, black or brown lines forming a network within the tissue. This contrast allows for the assessment of the structural integrity of organs. Diagnostic use. Retic staining is essential in diagnosing conditions like liver cirrhosis, myelofibrosis, and certain tumors, where changes in the reticular network can be indicative of disease. Trichrome staining. Purpose and application. Trichrome staining is a group of techniques used to distinguish between different tissue components, particularly collagen, muscle fibers, and cellular elements. It is widely used in pathology to assess fibrosis, scar tissue formation, and the overall tissue structure in various organs. Principle: The Trichrome stain utilizes a combination of dyes that selectively bind to different tissue components. Nuclear stain, a hematoxylin stain, is used first to color the cell nuclei blue. Acid dye, the tissue is then stained with an acid dye, such as biobrick scarlet, which colors cytoplasmic elements red. Collagen stain, finally, a collagen-specific dye, such as aniline blue or light green, is applied. This dye colors collagen fibers blue or green, depending on the specific trichrome method used. Key features. Visualization. The trichrome stain produces a multicolored image where nuclei appear blue, muscle fibers and cytoplasm appear red, and collagen fibers appear blue or green. This multicolor contrast is particularly useful for assessing tissue structure and fibrosis. Diagnostic use. Trichrome staining is crucial in evaluating liver biopsies for fibrosis, assessing myocardial infarctions, and studying connective tissue disorders. The human touch in staining techniques. While the science behind ZN, Retic, and Trichrome staining is well established, the human element is critical in their successful application. Laboratory technicians and pathologists must possess a deep understanding of the tissue types they are working with, as well as the specific diseases they are trying to diagnose. The precision in preparing tissue sections, the timing of each staining step, and the careful interpretation of the results all require a skilled and experienced hand. Moreover, the ability to troubleshoot and adapt protocols when faced with challenging samples or unexpected results is a testament to the expertise and judgment that human professionals bring to the process. Each stained slide is not just a result of chemical reactions but also a reflection of the dedication and care of the laboratory staff. Conclusion. ZN, Retic, and Trichrome staining techniques each serve unique and vital roles in the field of histology and pathology. ZN staining provides a reliable method for detecting acid-fast bacteria, crucial for diagnosing infectious diseases like tuberculosis. Retic staining reveals the delicate reticular networks that support various organs, aiding in the diagnosis of structural abnormalities. Trichrome staining offers a detailed view of tissue composition, allowing for the assessment of fibrosis and other connective tissue disorders. These staining techniques, while rooted in scientific principles, are also deeply influenced by the human touch. The expertise and precision required to carry out these methods ensure that the final results are not only accurate but also meaningful, providing critical insights that guide patient care and advance our understanding of disease.